Nutrients are the chemicals that provide energy and building block for new substances within the cell. Microbes have to obtain the nutrients from their environment. Then they have to be brought into the cell. The cell membrane is the barrier to this. That is why there are transport mechanisms. All cells, both prokaryotic and eukaryotic, use diffusion, facilitated diffusion, and pumps. Prokaryotic cells use group translocation, and eukaryotic cells use endocytosis. For microbial growth to occur, all organisms have to have a carbon source and an energy source. Additionally, they have to have a source of hydrogen atoms because hydrogen atoms are involved in those redox reactions. And then oxygen and nitrogen requirements must be met, plus a few trace elements. We can divide microorganisms up into nutritional types based on their source of carbon and energy. Photoautotrophs use carbon dioxide as their carbon source and light as their energy source. Photoautotrophs perform photosynthesis, so plants, algae, and the cyanobacteria are photoautotrophs. Anything that makes its own food is not going to be pathogenic. Chemoautotrophs use carbon dioxide as their carbon source, but they get their energy from inorganic chemicals. These are things like lithotrophs and methanogens. Lithotrophs actually break down rocks. Methanogens make methane gas. The hydrogen, sulfur, and nitrifying bacteria fall into this category, as do some of the archaea. Again, anything that makes its own food is not going to be pathogenic. Photoheterotrophs use as their carbon source organic molecules, but they're capable of using light as their energy source. There are a few bacteria, the purple and green bacteria, and some archaea that can perform this type of metabolism. Again, with light as their energy source, they're not a source of pathogenic organism. The chemoheterotrophs use organic molecules as their carbon source, and they use organic molecules as their energy source. They break down organic molecules to provide the ATP. They also use the organic molecules as their source of carbon. This type of organism is sometimes called an organotroph. We are chemoheterotrophs, as are the pathogenic bacteria. Organisms have different oxygen requirements. Obligate aerobes must have oxygen. They require oxygen to live. Oxygen is the final electron acceptor in the electron transport chain they use. Obligate anaerobes find that oxygen is toxic to them. They lack the enzymes to inactivate the toxic forms of oxygen. And basically, there are 4 oxygen molecules that are toxic. Singlet oxygen, which is single oxygen, not O2, superoxide, peroxide, and hydroxyl. Most facultative anaerobes and obligate aerobes have enzymes that break these down so that these are not toxic. All of these molecules are potent oxidizing agents. That is, they capture electrons, so they oxidize other substances. Facultative anaerobes are capable of either aerobic or anaerobic respiration or fermentation. They don't require oxygen, but oxygen is not toxic to them. A few organisms are what we call aerotolerant anaerobes. They do not use aerobic respiration, they do not use oxygen as the final electron acceptor, and they can tolerate oxygen for brief periods. They have some enzymes that will detoxify the toxic forms of oxygen, but not many. Microaerophiles require a reduced oxygen content in their atmosphere. Many of them are what we call capnophiles. They like reduced oxygen, but they like an increased carbon dioxide atmosphere. The nitrogen requirements for organisms are pretty readily available. Proteins are the biggest source of nitrogen in organic molecules, and then there are all sorts of forms of inorganic molecules. Nitrogen can be a growth limiting factor. If there's not enough nitrogen in the environment, metabolism will stop because the organism will be unable to make proteins, and proteins are required to make enzymes and structures within the cell. Other chemical requirements include things like phosphorus, which is required to make nucleotides, and sulfur, which is required for some amino acids. Then there are trace elements like calcium, manganese, magnesium, copper, iron, zinc, and a few others. These are usually dissolved in water. However, these too can be growth limiting. Some organisms require growth factors. Growth factors are organic molecules, mostly vitamins. Organisms that require this kind of growth factor are usually considered fastidious organisms. If we limit any nutritional requirement, we can control the growth of any organism. In addition to chemical requirements, organisms have certain physical requirements: temperature, pH, osmotic pressure, and hydrostatic pressure. Each organism has an optimum requirement for each physical requirement, and it will grow best when all conditions are at their optimum. If we remove optimum conditions, this is another way we can control growth. Temperature can affect enzyme structure and metabolic rates by speeding up or slowing down chemical reactions. The minimum growth temperature is the lowest temperature at which metabolic processes will occur in the cell. If we go below this, we may kill the cell, but more often we simply suspend growth. The maximum growth temperature is the highest temperature at which metabolism occurs. If we go higher than the maximum temperature, this usually denatures proteins, and this will kill the organism most of the time. The optimum temperature is that temperature at which the organism's metabolic processes will function at the highest rate. At the optimum temperature, we will see maximum growth of the organism. Organisms can be classified based on their preferred temperature range. Psychrophiles grow best at about 15 degrees centigrade. They can grow below 0 degrees centigrade, but usually die at temperatures above 20 degrees centigrade. While these organisms are important in food spoilage, they are typically not pathogenic. The process of lyophilization, or freeze-drying, is actually a means to preserve cultures. Mesophiles have an optimum temperature range between 20 degrees centigrade to 40 degrees centigrade. This is where the pathogens are because body temperature is 37 degrees centigrade, right there in that range. Psychrotrophs are mesophiles that will grow below 20 degrees centigrade. These organisms are sometimes involved in food poisoning because they will multiply at refrigerator temperatures, and if they produce toxins, the toxins can be produced in the food. Thermoduric organisms are mesophiles that can survive brief periods above 40 degrees centigrade. These organisms sometimes are also involved in food poisoning because with the microwave oven, we frequently don't heat food high enough to destroy some of the toxins produced by microorganisms. Thermophiles are those organisms that grow at above 45 degrees centigrade. Hyperthermophiles actually will grow in water above 80 degrees centigrade, and we have a few archaebacteria that live at temperatures above 100 degrees centigrade. Most of the thermophiles are in the domain Archaebacteria. Most organisms grow best in a fairly narrow pH range. Neutrophiles prefer a pH between 6.5 and 7.5. Blood pH, you'll remember, is 7.35 to 7.45. Acidophiles live in a pH between 0 and 4.5. Some organisms are obligate acidophiles. They require an acid environment to grow. Other organisms produce acid end products like lactic acid, so they need to be able to tolerate the acid conditions they actually produced. A few organisms are acid tolerant. They can survive an acid environment, but they do not thrive in it. Alkalinophiles have a pH range of 7 to 10. We do have a few organisms that will grow in environments as high as a pH of 11.5. Vibrio cholera, the organism that causes cholera, actually prefers to grow at a pH of 9. It really grows best outside of the body. All cells require water, and water moves into and out of cells by osmosis. If we put cells in a hypotonic solution, the cells will swell. If we put them in a hypertonic solution, the cells shrink and literally dehydrate. Most cells have capsules, slime layers, or substances in their cell wall like mycolic acid to help prevent drying. There is a group of organisms called halophiles. They grow under high osmotic pressure. If they are obligate halophiles, they have to have this high salt environment. Some of them grow in salt solutions as high as 30% salt. They require this level of osmotic pressure to live. They will even die if they are not transferred under high salt conditions. Other organisms are facultative halophiles. They can withstand high salt environments and grow in them, but not grow well. Then we have barophiles. We have some organisms that have been found very, very deep in the ocean. There's a lot of hydrostatic pressure above them, and they require this high hydrostatic pressure in order to grow. Environmental associations between organisms can include antagonistic relationships where the two organisms do not get along. The one we'll be studying will be the antagonistic relationship of parasitism. One organism thrives, but the other organism is harmed in this relationship. There are beneficial relationships. In a synergistic relationship, both organisms benefit, but both organisms can live separately. In a symbiotic relationship, the organisms become dependent on each other and cannot live separately. Biofilms are complex relationships among numerous organisms. Most organisms in nature live in a biofilm, and biofilms colonize or cover a great many pieces of equipment in hospitals. The CDC estimates that about 70% of disease is actually caused as a result of biofilms. In a biofilm, the cells will communicate with each other and coordinate their activities. In this sense, they act similarly to a tissue. A biofilm will begin to develop when free-living cells settle on a surface. These cells develop a gooey matrix that's composed of DNA, proteins, and tangled fibers of the glycocalyx of the cells. The cells stick to each other and the matrix. This helps them sequester nutrients and form water channels. It also provides them with protection from environmental stresses like changes in temperature, pH, the presence of drugs or disinfectants. As the organisms develop, they start producing quorum sensing molecules. Microbes begin to respond to the density of nearby microbes. These quorum sensing molecules are secreted and they act to communicate the number and types of cells that are in the biofilm. Many cells will possess receptors for quorum sensing molecules. As the density increases, so does the concentration of these molecules. Once the binding of microorganisms exceeds a certain threshold number, the microorganisms in the biofilm begin to express previously suppressed genes. Microbes now will display new characteristics. They will produce enzymes that they've never produced before, they will start to change their cell shape, and other changes will occur. The matrix also allows the members to concentrate so that they can conserve their resources. Instead of digestive enzymes diffusing away, they stay in one area, and more organisms benefit from fewer digestive enzymes, for example. New microorganisms can constantly arrive, and new synergistic relationships can develop in the biofilm as the biofilm organizes into a community. The individuals of the biofilm display metabolic and structural traits that are different from those expressed when the organism lives independently. The members of biofilms take on different roles in different parts of the biofilm. Some individual cells or groups of cells are capable of leaving the biofilm. These are called streamers. Many organisms become more harmful when they're in biofilms because they start secreting toxins that they were unable to secrete before. Dental plaque is one of the most common biofilms, and it's involved in tooth decay. Because biofilms are such a problem in hospitals and other health-related areas, they're trying to prevent the production of biofilms. One of the ways they are trying to prevent the development of biofilms is by using drugs that block the receptors that are involved in the production of biofilms. Now here you see a few organisms free in the environment that settle on a surface. The cells then begin producing the matrix, and quorum sensing molecules begin to be released. This changes the biochemistry and the shape of many of the cells in the biofilm. New cells begin to arrive. New species arrive. We have water channels forming. From time to time, some molecules escape as streamers. They may go and form a new biofilm, or because they're now expressing different genes, if they get into a human being, they may cause a disease. You'll remember that bacteria reproduce by binary fission. The generation time is the time for one cell to grow and divide. This varies greatly from organism to organism. Under optimum conditions, the generation time is the shortest time possible. Under optimum conditions, E. coli's generation time is 20 minutes. That means if you start with one cell, in 20 minutes you'll have two, in 40 minutes you'll have four, and so on. The generation time for something like Mycobacterium tuberculosis is more like 10 hours. We can look at the phases of microbial population growth by plotting a growth curve. That is, we look at the number of organisms present based on how long the culture has been growing. We will see four distinct phases. The lag phase is the first little bit of the growth curve. This is when the organism is adjusting to the environment. If there's a new nutrient present, the organism may have to start producing a new enzyme in order to process it. During the log phase, the cells are actively dividing. Every generation, the population doubles. Log is for the phase of logarithmic growth. After a while, the cells that started in the culture begin to die. However, cell division keeps going and we keep the population at a stable number. After a while, if we've not changed out the nutrients, toxins begin to build up. This helps kill off cells, and cells begin to die faster than they are reproduced. Looking at this growth curve, this is the number of live cells over the time. Short lag phase, a log phase when we're doubling every generation period. Then we go into the stationary phase. This is when as many cells are dying as are being reproduced. After a while, if we don't change out the nutrients, we will begin to have more cells die than are reproduced. Now, we never get down to totally zero cells alive, or we don't for a long period of time. This is a very shallow curve. So even after a few weeks, you can go into a culture that should have been in this death and decline phase for a while and still find viable cells. A continuous culture can be done if we keep refreshing the media. That death phase occurs simply because the organism builds up toxic waste products and runs out of nutrients. A chemostat is an open system where the media is constantly replaced. You can use a media that has limiting growth factors so that you maintain a steady number of organisms, or you can maintain microbial populations at very low nutrient levels. A chemostat can also be used to study organisms in the log phase. If we want to measure microbial reproduction, there are several techniques we can use. Direct cell counts can be done, and incubation is not required here. You can actually look at cells on a microscopic slide using a very special slide that has a grid on it and literally count the number of cells. There are also electronic counters that can be used. There are direct methods that require incubation. One of these is the serial dilution method and the making of pore plates. This is particularly helpful if you have a large population of microorganisms that you want to study. Membrane filtration is good if you have a small population of organisms in the media. You simply filter the solution and then culture the filter and see how many colonies are produced. There are indirect methods. One is turbidity. You can measure this in a spectrophotometer to see how many organisms are present. You can measure the use of nutrients out of a culture and base it on metabolic activity. You can filter the organisms, dry them, and weigh them, and calculate the approximate number of cells that are present. Or you can look for genetic methods. Remember, we've said that most prokaryotic cells have yet to be cultured, but we find them in the environment. If we find a DNA sample and then look for how much of that DNA is present, we can determine approximately how many organisms are in that particular environment.